Hi everyone, my name is Emily-Anne Hicks and I'm a PhD student in Heather Sheardown's lab at McMaster, and I'm going to give you a quick overview of my project, which is exploring the role of lubricin in retinal pigment epithelial cell homeostasis. So what is lubricin? It's an extracellular matrix protein which is well known for its lubricating properties. It reduces shear stress at the surface of articular joints via its heavily glycosylated mucin-like domain. However, with near universal expression in adult tissues, PRG4 is no longer solely this lubricating protein. It has become associated with a variety of physiological responses, and that includes regulating inflammation. So lubricin acts as an anti-inflammatory agent by coating immune cells and non-immune cells like fibroblasts and epithelia through its interaction with a variety of surface receptors, and that includes CD44, Toll-like receptors, and L-selectin. And by coating these receptors, lubricin can buffer the cell's response to pro-inflammatory mediators. In a dry eye study, the loss of lubricin expression from ocular surface epithelial cells results in this inflammation-based damage that you can see with an increase in corneal fluorescein staining. And by adding lubricin back to the eye in the form of eye drops, the researchers observed a decrease in this corneal staining when compared to commercially available eye drops. And my preliminary work shows that not only is lubricin in the cornea, but it's actually throughout the eye, including the retinal pigment epithelium. And I've shown in Western blot analyses that lubricin is upregulated in human RPE cells when exposed to a stress condition, similar to what was just previously shown in the front of the eye. And so the goals of my PhD are to understand the homeostatic role of lubricin in the RPE cells under stress conditions. So essentially how lubricin regulates the inflammatory response in the back of the eye. And then looking at the efficacy of lubricin as a therapeutic for AMD, essentially. So this would be done with in vitro and in vivo models of oxidative stress and choroidal neovascularization. And then lastly, I want to look at the long-term delivery of lubricin to the back of the eye. And this would be done with pigment gels, and that would just be to prevent the degradation of lubricin upon injection. And why I'm doing this research is because the RPE is a highly specialized epithelia, and it is essential for photoreceptor health, which is crucial for maintaining your vision. And oxidative stress and inflammation are both reported factors in age-related macular degeneration pathophysiology, and they both negatively impact the health of the RPE and then by extension, the overlying photoreceptors. And if lubricin is able to regulate the response of these cells to oxidative stress and inflammation, it could prove to be a useful treatment for AMD, specifically dry AMD, which currently has very little treatment options available. And that's just a quick overview of what I'm hoping to do. And if anyone has any questions or wants to know more, feel free to reach out to me. Thanks.